Now we're going to talk about the nucleus and the structures inside of the nucleus, the functions that occur within the nucleus. So the nucleus is the largest structure inside of the cell, and within the nucleus we have all of our DNA that makes up the blueprints of who we are. Including inside the nucleus are tiny structures called the nucleolus. So this structure here is called a nucleolus, and some cells have multiple nucleoli. We do have some cells that do not contain any nuclei. So red blood cells are an example of a cell type that has no nucleus. And the reason for this is because the red blood cell's function is to carry oxygen. And for it to do so effectively, it basically fills its entire cell structure up with hemoglobin. And hemoglobin is what binds oxygen. And then there are some cells that have more than one nucleus. So cardiac muscle fibers can have up to 2 nuclei. The skeletal muscle can have multiple nuclei. The nucleolus then is the tiny little structure inside the nucleus, and this is where ribosomes are produced. Ribosomes are composed of proteins and RNA, and once the ribosomes are produced, they then leave the cell. The small and large subunits leave the nucleus, not the cell, nucleus through the nuclear pores, and they enter the cytoplasm. So there's an actual figure of the nucleus. This is using a transmission electron microscope, and this is 20,000 times larger than what it actually would be, the size of it. So that's huge. DNA is what is found inside the nucleus. So DNA is found in the nucleus outside of the nucleolus. And it basically takes up the majority of the room in the nucleus. DNA is composed of 4 nucleotides: adenine, thymine, guanine, and cytosine. DNA is the polymer. The monomers are the nucleotides. They're bonded together through phosphodiester bonds, and then because DNA is a double helical molecule, it has 2 strands. The strands are held together, or the nucleotides are held together in the middle by hydrogen bonding. So the phosphodiester bonds, which form on the backbone and hold the DNA together, or hold the single strand of each side together, is formed by dehydration synthesis, and it binds sugar and phosphate, sugar, phosphate, sugar, phosphate. The sugar is called deoxyribose, and then we have a phosphate. That makes up the sides of the ladder. And then in between the nucleotides, we have hydrogen bonding. So that's what these are. Between guanine and cytosine, we have 3 bonds. Between thymine and adenine, we have 2 bonds. Adenine and thymine always pair together. Guanine and cytosine always pair together. DNA in its loose or relaxed form is called chromatin, and I always think of chromatin as kind of like spaghetti. So there you have like this bowl or nuclear bowl full of the spaghetti noodles, and then when we are going to divide, when the cell's going to divide, the DNA condenses, and it condenses using these little proteins called histone proteins. These histone proteins are kind of like the meatballs of the spaghetti, and the DNA winds up around those histone proteins, forming a chromosome. And this is what most people think of when they see DNA. So it only is condensed when we're dividing. Otherwise, it's always in its uncondensed form, and that's how we can read the DNA to produce proteins. And so there's another figure showing the condensed formation of DNA. So I told you that DNA is the blueprints of who you are. It helps to make up your the color of your eyes, the pigments of your skin and hair. It makes up the skin itself. It makes up your muscles. It makes up your organs. I mean, so proteins make up everything. And DNA carries the blueprints for the proteins that make up everything. DNA is the blueprints of protein synthesis. And so it carries the information that makes up proteins or genes. So here we have this gene. Okay, some gene. It could be a gene for insulin. It could be whatever you want to call it. There's always going to be a promoter site, and there's always a terminator site. And then we have the actual structural information in between. The first part of producing a protein is called transcription. This is where DNA is transcribed into a language that our cells can understand. So DNA isn't usable by the cell as a language. It's basically a language that we don't understand. So we take DNA and we produce a language that we do understand in the form of RNA. So it'd be like, think of a language you don't know. So most people aren't fluent in like Latin. That's not a language we're all fluent in. And so, say the DNA was a Latin manuscript. Now, we don't understand it. We can't read it. So we can't produce the whatever functional thing that manuscript's trying to tell us to produce. So we take the information of that manuscript that's in Latin, we transcribe it to a language we understand, say English, and now we can produce whatever is written in that manuscript. So we transcribe it from, in this case, DNA to a language our cells understand, which is RNA. And then once we have transcribed it, we then can translate it into the proper protein. And that is protein synthesis. So for transcription to occur, we have to have the DNA that codes for whatever protein we care about. We have to have nucleotides that will produce the RNA form of the DNA. So these would be called ribonucleotides instead of deoxyribonucleotides. We have to have ribose sugar and then the phosphates. And we have to have the enzymes to activate RNA synthesis, which would be RNA polymerase. So there are 3 types of RNA that we actually produce through the process of transcription. Messenger RNA is the one we typically focus on because it's the RNA that we then translate into a protein. But we also produce transfer RNAs, which are a form of RNA that can carry amino acids. And so remember that proteins are the polymers and amino acids are the monomers that make up the proteins. And then we have ribosomal RNA. This is the functional ribosome. Our ribosomal RNA, add protein to it, you produce the functional ribosome. And the functional ribosome is what, or is where proteins are produced. So it's like the workhorse. It runs along the messenger RNA and it produces the protein. So for transcription to occur, we have 3 phases or steps. Initiation, where the RNA polymerase binds to the promoter region. Elongation, where the RNA polymerase runs along the DNA and allows the proper RNA nucleotides to come in. And then termination, where we hit that terminator sequence and the DNA closes back up, the RNA polymerase leaves, and the RNA that was produced can then be modified. So initiation, we start at the start or the promoter sequence. We're going to bind, and then RNA polymerase binds, and then we can start producing an identical copy of the coding strand from what we call a DNA template. So if this is our coding strand, this is the strand that has the gene for insulin or whatever gene we care about, right? We use the template strand, the opposite strand, as our actual coding strand. Or not as our coding strand, as the strand that we're going to have our RNA polymerase bind. So RNA polymerase binds, and then we produce a copy of RNA from this template strand. This copy of RNA is going to be identical to the coding strand. And so here where the T is in the DNA and A binds. In RNA, so RNA, remember, it differs from DNA in that it's always single-stranded and there's no thymine. When thymine would be present or would bind, you have uracil. So wherever we have an A in the DNA, the complement U will attach. G to C, G to C, A to U, and then wherever there's a T, you're going to have an A. And so we're just producing the identical copy. The only difference is in this messenger RNA and this coding strand is wherever we had a T up here, we have a U. And so look, A, T, U, C, 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 T, U, G, 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 T, U, A, G, G, A, T, U, C, 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 T, U, T, U, A, G, G. Do you see how this is identical to this except for where there's T's, we now have U's. That's the only difference. And so this really shows how transcription works and why we use the template strand to produce our messenger RNA and not the coding strand. Elongation occurs once RNA polymerase binds, then it runs along the DNA until it hits that terminator sequence. And so this allows the entire gene to be transcribed. And then we hit the terminator sequence, the RNA polymerase leaves, the DNA will start winding back up. The hydrogen bonds between the DNA and the RNA are going to break, and the messenger RNA can now be modified. And so this is your transcription. I don't know what this is. I may play it, I may not. Really depends on what it is, because I was like, well. I have a lot of good songs, but I want to make sure this is the one I want to play at any given time. No, this is not a song I want to play now. That's for replication. So, yeah, we're not going to play that. Sorry. All right. We'll play it later, though, so don't worry. So, now that we have our messenger RNA, we have to modify it before it can leave the nucleus. So I always like to ask this. I ask this in my AMP class and my micro class, and what's kind of funny is typically when I ask it in my AMP class, my students, aren't sure of the answer. They kind of guess. And if they're in my micro class, again, they either forget the answer, or they're too scared to answer because they think that maybe they are wrong again. But I do ask, so if you ever take my micro class, you'll probably hear some of the questions I ask now. So the initial messenger RNA is considered premature messenger RNA. It's not going to be released into our nucleus, and there's different reasons for it. The first is pre-messenger RNA. If it leaves the nucleus, it's going to be degraded very rapidly. So we have to add some nucleotides to the ends and to the beginning. But also, I like to ask this. I'm going to ask, and then you can think about it, but I'm not going to give you the answer. So you have to bring this to class and ask me about it, okay? How much of your DNA do you think codes for proteins? The only thing I'll say is it's not 100%, and it's obviously not 0%, but it's somewhere between the 0 and the 100, okay? So that's what I'm going to say, and now we're going to show you what happens. So we're going to modify the DNA, and the first thing we have to do is all the regions of our DNA that don't code for proteins, these are called introns. And I think of introns as commercials. So when you're watching a good movie, a really great movie, and you're right at one of those exciting parts where you know something's going to happen, the music gets heavy, and then all of a sudden a commercial comes on. The commercial doesn't have anything to do with the movie, usually. And it's usually the time where you run to the bathroom, you get another glass of milk or water or whatever it is you're drinking. You go grab some snacks, whatever it is you're going to do. And then you get back in to watch the next part of the movie. So the regions that you're watching, or the regions of the DNA that coded for proteins, are called exons. The non-coding regions, which I call the commercials, are the introns. And so we have to get rid of all of the introns, the stuff that doesn't code for the protein, and then we have to splice together all of the exons. And so we cut out all the introns, and we put together all those exons, and we now have a functional messenger RNA molecule down here. But we have to add a poly A tail and a guanine cap. The guanine cap protects the DNA. So in our cytoplasm, we have enzymes that will degrade DNA or messenger RNA. It'll degrade nucleotides or nucleic acids. So we add the guanine cap to prevent the nucleic acid from being digested too quickly. It will get digested, but it won't get digested so fast that we can't first translate it or produce the protein. So we do that. We add the poly A tail for the same reason, but we also add the poly A tail to protect the ribosomal RNA from falling off too early. So as the DNA, or as the ribosome attaches and starts moving along this molecule, this long messenger RNA molecule, when it gets to the very end where you have some stop codon, ribosomes are large, and so they're going to sit on here, and as they're moving, if they get to the end too fast, the ribosome can actually fall off, and you can miss one or two amino acids. So that would be a deletion of amino acids, and that can cause different disorders. And so the poly A tail helps to keep the ribosome on until you hit that stop code that stops translation. All right, so I've talked for 20 minutes. I'm going to stop this video, and I'll get translation in our next video, okay? All right, I'll chat with you in a few minutes. Bye.